You know, one of the things that I do is I check in with scientists that we've talked to. Some of them are behind the scenes, others decide to come forward at different times, but with questions. What would the questions be when it comes to the coronavirus vaccine? There are obvious questions that they're already admitting that they haven't answered. Things like what a vaccine does, which is stop transmission. They don't know if it does. I don't even know how this thing can be called a vaccination until it proves that it can stop the infection and stop transmission. But science doesn't seem to be working the way it used to, where we were asked hard questions, where we challenged the theory, where, actually checked in with, I don't know, maybe heart doctors and neuroscientists to figure out what they thought. Well, we've done a lot of reporting on the immune system and the potential for autoimmune issues when it comes to the COVID-19 vaccine. We've discussed, fertility with scientists and doctors that can discuss that issue. But I was talking to a neuroscientist that has done some work in this field that brought us a lot of the studies on the dangers of aluminum in vaccines. Of course, I'm talking about the world-renowned scientist from the University of British Columbia, Dr. Christopher Shaw, and it is my honor and pleasure to have him joining us right now on the Highwire. Chris, you brought up something. I sort of just stopped you the other day and said, you know what, I want you to bring that live on the Highwire. From a perspective of neuroscience, there's lots of concerns that causing the cell to create a spike protein in the body and hoping that the immune system somehow creates antibodies to fight that could start some cycle that could lead to long-term autoimmune illness. Now, of course, there's excuses, but not a lot of science has been done. But you have a particular concern when it comes to the brain. What is that concern?
Well, the concern is actually several-fold. One, I looked at the Pfizer and the Moderna phase 3 safety studies, and there were things that were disturbing in that. One of the biggest ones that I noticed was the Bell's palsy, and then I went back to the animal literature where some Moderna scientists had actually looked at where the construct, the mRNA lipid-coated polyethylene glycol construct had gone. And they were finding it in all kinds of parts of the body. And the official story is that, once you inject it into your muscle, it stays there, it gets into your cells, and the lipid helps you do that. And then it, attaches to the ribosomes, and it turns out this protein that your immune system will recognize, and all is good to go. The problem is in their own study, this is done in 2012, the first author is a guy named Kapil Bal. And what you see when you go to their table 2 is it looks like the construct, and this was done with a different virus. But it was the test of the mRNA vaccine in general. And as with the Pfizer and the Moderna stuff that came out with humans, they're getting pretty good immune response initially, it looks like.
Yep.
But they're also finding the construct in the brain and other places. So that tells us something that it's not breaking down as fast as it's supposed to. Remember, the whole basis of the cold chain storage is that this is a very labile molecule, that it will not survive. And somehow it is surviving to get inside various organs, including the brain. And that table tells you that this is maybe something we need to worry about, and we need to study in more detail, not just launch a population of 7 billion people, because I think you're inevitably going to have some consequences that we don't understand. So why is it there? It's not much compared to how much is in the muscle, but there shouldn't be any. So why is it there?
I want to bring up that really quickly, because obviously this looks like these were animal models, correct? And so they're able to kill the models after delivery of the technology, right? The mRNA technology.
That's right.
But folks, look at all the places they found it. It's like it blew up inside of the animal. We're talking bone marrow, brain, lymph nodes, heart, kidneys, liver, lungs. I guess if your goal was for it to travel all throughout the body, then mission accomplished. But what I guess my question is I've even seen doctors putting out videos that any concern that this crosses the blood-brain barrier are not true. It's been proven that that's not true. And for a layperson like myself, I ask myself, why are you even telling me that? First of all, what would be the problem if it does cross the blood-brain barrier? Why are they defending against that? So let me ask you that. What could be the possible problems should this mRNA technology make it into your brain?
Well, my first reaction would be that it could trigger some autoimmune reactions inside the brain, inflammation, because when you release either the protein, in this case it may be the mRNA itself, because it's inside the brain, you're going to trigger microglia, your resident immune cells, to do something about it. And what are they going to do? They're going to try and deal with the cells that are releasing it, and or wherever it is. Maybe they'll just engulf it and it'll be harmless. But the onus is on the company to actually show us that it's not harmless. And they started to do that back in 2012 with the mice study. But I would think that a lot more rigorous science needs to be done about this before you do a giant experiment with 7 billion people. And you don't want to be coming back 2 years later or 3 years later and say, oh my goodness, where did all this autoimmunity come from? And I think it's a concern. And I think it really is incumbent upon the companies and NIH and CDC and all those entities that monitor the safety of these things actually to commission some studies and say, where does it go? Because clearly, based on Moderna's own data, it goes into various compartments of your body, including brain. Again, not much, but how much is safe? We don't know. And they never followed it over time. The animals are sacrificed or after a few days after injection. So how much goes there eventually? Why is it going there at all? I mean, that doesn't make any sense based on what they're telling us about how easily degraded this molecule is. So it shouldn't be there. It is there according to their own data. Maybe they were wrong. Maybe they didn't do it right. But again, that calls for more studies, not fewer.
Absolutely. And so when you're saying autoimmune disease, leading to autoimmune disease in the brain, I always think of autoimmune disease sort of in the body. But you're saying that the glia cells up there that sort of handle immunity in the brain, when you say it could lead to inflammation, are you talking about is the potential for brain swelling? Is that something that could happen?
I don't know. I mean, again, I'm only going on this one study. There are a couple others that show somewhat the same thing. But when you turn your microglia loose against targets inside the brain, you're going to get inflammation of some sort, and it's going to start targeting cells. The other thing, as a neuroscientist, I work on neurodegenerative diseases, and what characterizes all of them, virtually all of them, is this misfolded protein that is usually characteristic to Lou Gehrig's disease, to Alzheimer's, to Parkinson's, to Huntington's. There are different proteins, but what they tend to do is they tend to form these unusual kinds of sheets of misfolded proteins called beta sheets. Now you're asking cells in various parts of the body, including possibly the brain, to make a lot of proteins and release it to the outside. Are we sure that's what it's all doing? Are you getting clusters of misfolded proteins inside neurons? That would be a bad thing to do. And you'd like to know where it is, how much of it there is, and which particular neuronal groups it's targeting. And those are the kind of questions I think you would easily have wanted the companies to solve long before they started authorization and discovering some years later that they have a problem.
What would be your advice? How long does it take to answer? I've had different scientists that are asking different questions. We've talked about the placenta, the potential attack of if there's immunity to a spike protein that resembles, the lining of the placenta. But for your concern, if you were to say, hey, Pfizer calls you, Moderna calls you, or Anthony Fauci says, you are, good at designing studies of the brain, how long would it take you? How would you design figuring out if we have an issue of the mRNA vaccine entering the brain? What would be the process?
You would, if you were doing it in mice or monkeys, that's essentially a discussion of cost. If you were doing it in mice, like they did this mouse study, and remember, it was ridiculously underpowered. They only did 3 mice per group. But you would probably want to do adequately powered 10, 15 mice per group, however many groups you want. And then you would do detailed histology and biochemistry of all those regions of the brain. You'd be looking at maybe a year's work, not much more than that. And so is it out of their budget? Probably not, with the billions of dollars they have. Could they easily do it? Sure, there are a lot of scientists who could do this. I'm not the only one. There are any numbers who could do this. And then you would have an idea, is this a problem or not? Because you'd not only do detailed analysis to see if there are changes that suggest impacts on particular brain regions, but you'd also be looking at the cellular level. You'd be looking at the biochemical level. And again, not hard. All those techniques take you maybe, I'd like to say a year, totally, maybe not even that much. And then you would have a much better idea. And the fact that they're not doing it, well, that's typical. They rarely do histology on brains in animal models run by the corporations. But here, especially when you're rushing it out the door, I think you need to do it. And I think it's not that hard to do.
Well, Dr. Shaw, I want to thank you for bringing that perspective. I think that we should be asking you. I think everyone assumes that science and scientists and people that care about humanity, people like Bill Gates, fund great scientists like you to make sure we've answered questions before we put people in harm's way. Is it your opinion that we have jumped the gun here, that we have rushed a technology into people before it's ready?
Yes, that is my opinion. I think this is a vast experiment that should have been done in the laboratory with animal models, and now it's being done on people. And we'll know the answer. And I think you're going to show some video clips in a little bit. We'll know the answer in some months and years to come. But the potential is that you're going to harm a lot of people while you're doing this experiment on humans.
Amazing. Yeah, really shocking. All right, well, I'm glad you are continuing to do the great work you're doing, asking the questions of science that should be asked. I wanted to thank you for taking the time, Chris, and we'll look forward to seeing you again sometime in the future, especially if we start seeing some issue with the brain, right, in the people that are getting this.
Well, let's hope it's not. But again, let's hope that it's not as serious as it might be. But again, the only way to find out now is we're going to watch the results emerge as people get the shots and as they begin to be monitored in more detail.
All right.
But again, that should have been done a long time ago.
Absolutely.
My pleasure, Joe.
All right, take care. If you like that clip, then be sure to check out our live broadcast of the Highwire every Thursday morning at 11 a.m. Pacific time. You can watch it on Facebook, YouTube, iTunes, and Twitter. We'll see you there.